Today I'm presenting my talk, which is called Batman, Fast and Accurate Integration of Single-Cell RNA-seq datasets via minimum weight matching. So the outline of my talk is the following. I'll go briefly over single-cell RNA-seq, just in case if somebody is not familiar with this set of technologies, and then I'll present the problem of batch effects, give the definition, example, and formulate the problem. And then I'll talk about the current approaches. There are a bunch of methods, and even state-of-the-art methods have their issues. I'll talk about my approach, which is called Batman, and I'll explain the acronym. Finally, I'll go over the evaluation metrics and how we compare Batman against other tools. Okay, so single-cell RNA-seq is basically a set of technologies that are used to profile gene expression on the level of individual cells. And currently, the state-of-the-art approaches are like 10x genomics, if we're interested in 3' end profiling, or SMART-seq2 in case if we want to profile gene expression on the using full-length transcripts. So in single-cell RNA-seq, we can think about each cell as an n-dimensional vector, where n is the number of genes. And the value of the element at index I is the expression of a particular gene I. So single-cell RNA-seq data sets, that's basically nowadays is so-called large, it's large-scale data sets, and they usually have in one experiment tens of thousands of cells. In current experiments, people are dealing with millions of cells. We can mention here Human Cell Atlas and many other data sets. So the number of genes is approximately 30,000 for human, and the data is very sparse. We usually have a lot of zeros, which we are referring to as dropouts. So usually when we work with single-cell RNA-seq, we use top 1,000 to 5,000 highly variable genes. So here in the picture, we see a t-SNP plot. That's basically a graphical representation of the data sets. So a nonlinear dimensionality reduction method. So frequently what we want to do with, single-cell RNA-seq data sets is to merge them into one. So for example, we have a lot of cells in our sample, and we cannot sequence them all at once in one run. So, we can sequence them in several runs, and then we are interested in merging them into one data set. Or we want to combine, for example, two data sets that come from different labs, but share, the same biological condition, for example. Or we want to combine data sets across different platforms. For example, 10x or SMART-seq2, they have different properties, the technologies. So therefore, we will see some discrepancy between two data sets. So we would like to, come up with a method that will be able to, merge two data sets into one, so that we have, basically more, powerful results in downstream analysis. So the main question here is how to correct, two data sets. Can we simply merge them, or do we need to do any kind of processing? So, okay. So first, let me talk about what batch effects are. So these are differences that we see due to technical variability. So technical variability comes from high level of noise in single-cell RNA-seq and stochasticity of dropouts. So we can visualize this by using PCA or UMAP t-SNP plots. So for example, here in this picture, we see two batches or two data sets that come from the same biological condition, but if we plot them using, let's say, top two principal components, we will see that there is, some discrepancy between them. So we would like to correct that, and at least visually, see that they are kind of well mixed with each other. So for correcting this batch effects, we have a bunch of existing methods. So I'll talk about them later, but first let me tell about how do we quantify batch effects. So is it simply looking at the picture, or maybe there are some other more appropriate methods? And the state-of-the-art approaches for quantifying batch effects, they're all based on basically matching or basically looking at the you know, like batch label distribution, like the global one and the local ones. So one of the approaches is KBAT, and here I'm presenting in this picture KBAT. There is another approach called LISI, which is local inverse Simpson index. But the idea is that, you know, like we first want to, you know, like compute the global batch label distribution, and then, you know, like the data sets are considered well mixed, or they are considered not having batch effects, when in each of the neighborhoods in the data set, you know, like we observe kind of similar batch label distributions as in the, you know, like global distribution. So for example, here in the first picture here, we see that, you know, like basically in each of the small neighborhoods here, the distribution of batch labels is kind of similar in statistical sense to the global distribution. Whereas in this picture here on the right, we see that, you know, like we have some, you know, like some neighborhoods where some of the data sets are not represented. So, and we like visually see that, you know, like there are still a lot of, you know, like bad neighborhoods, and the data sets are not well mixed. So that's a kind of, you know, approach to measuring it. LISI uses a bit different formula for that, but the idea is all the same. You know, like we need to have well mixedness of the two data sets. So given this kind of, you know, approach to measuring batch effects, our question is whether, you know, like single cell RNA-seq integration is a trivial problem. So one might think that, you know, like we can take two data sets, like for example, we have here two batches, batch one, which is blue, and batch two, which is orange. So, you know, like given that definition of well mixedness, can we just simply, you know, like take, for example, data set one, and then to each of the cells we can assign some, you know, like some vector, some shifting vector, so that we randomly shift each of the cells here, so that, you know, like they approach their counterparts here in the batch two. So you know, the problem with this approach is that, you know, like we just, you know, compromise the local structure of each of the data sets. So we are destroying, you know, the cell-to-cell relationships. So that approach is basically wrong. So here we also have to take into account, you know, like local structure of each of the data sets. So current approaches for batch effect correction, they can be subdivided into two you know, like main types of approaches. So the first one, they, you know, like they operate in the gene space. So in this case, we have, you know, like multiple problems like curse of dimensionality. You know, like when we have, like, let's say thousands of dimensions, even if we're using only highly variable genes, which are, you know, like 1 to 5,000. Even in this case, there is a high impact of this curse of dimensionality. And it's also a lot of, you know, like computational burden to use, you know, like data sets in the gene space. The good thing about this is that we can use, we have, you know, afterwards we have a broader range of downstream analysis. We can use the data sets, which are, you know, batch corrected data sets in differential expression analysis, and so on and so forth. So examples of such methods are MNN correct, Seurat version 3.0, and Scanorama. And there are methods which operate in the latent space. They usually use, you know, like PCA or CCA or any other, you know, like even nonlinear embeddings. Like, for example, SCVI is using, you know, like neural networks to generate, you know, like latent space representations. But the problem here is that, you know, like the range of downstream analysis is kind of limited, since we cannot, you know, like use the values, the batch corrected, you know, expression values in, let's say, differential expression analysis. So, okay. Now the main paradigm, I'm talking, so from here and, you know, to the end of my talk, I will be concentrated on the, you know, on the methods that operate in the gene space. And so my goal was, you know, developing a method in the gene space. So the main paradigm of gene space methods is so-called mutual nearest neighbor. So here I'm, you know, like I'm showing you a picture from MNN correct method, and I'm talking about the main idea here. So the main idea is when we have, for example, two batches, batch one and batch two, right? So we want to, or like this method identifies, you know, like a pairs of cells. So where one of the cells belongs to batch 1 and the second cell belongs to batch 2, such that they're mutual nearest neighbors. So this means that for each of the cells in batch 1, we find, let's say, top k nearest neighbors in batch 2, and the same for batch 2. For each cell, we find top k nearest neighbors in batch 1. Now, when we have this kind of mutual relationship here, so we say that this pair of cells is mutual nearest neighbors. So this, pairs of cells, they're used to, to guide the integration process. So these are so-called like anchor pairs. So that's the main approach. The issues that were identified by even by the authors of the paper is that, eminent correct approach is kind of vulnerable to large batch effects, or when the batch effects are non-orthogonal to the biological signal. I'll talk about this and how, about these problems later in my slides. So basically, my approach here, that I'm proposing is called BATMAN, and the acronym stands for Batch Effect Correction via Minimum Weight Matching. So basically, one of the features of this eminent approaches is that, the mutual nearest neighbors, when we actually do the, visualization of this anchor pairs, we will see that they generate anchor pairs basically on the frontiers of the two data sets. So this means that, there is so-called kissing effect. And, this thing is kind of good for cell population matching when, when we want to basically maybe transfer labels from one data set to the other one, but it's bad for a proper correction of batch effects, since, it will create the situation when the batch effects will be, undercorrected. So the idea that I came up with in order to overcome this kind of issues, is the following. So we want to not match only mutual nearest neighbors, but use them to guide the integration process. But we want to use cells throughout the whole volume of the two data sets, and we also want to ensure that the move of the points is a coherent one. So the approach that I'm proposing here is basically is a parsimony approach. So here in BATMAN, let me tell you what's the global idea here. So the global idea is that we want to okay, first let me start with the definition. So suppose that we sequence a cell twice in the context of two data sets. So we have two batches, batch A and batch 2, and let's suppose that we have a cell that we could have sequenced twice. That's obviously impossible, but then what we would have, we would observe, what would be the measurement or the gene expression of that particular cell in the context of dataset A, and then in context of dataset B, or dataset 1 and dataset 2. Now, what we could do, by using the same cell, we could estimate the batch effect vector. Obviously, this is not possible because when we sequence the cell, we destroy it, right? So now, suppose that for each cell, the assumption is that for each cell in the first data set, there exists such a sibling cell in the second data set, which would be, the closest to its representation in case if we could sequence it twice. So the formulation of my approach is basically the parsimony is finding an alignment of the first data set to the second one, such that the total length of the estimated batch effect vectors is minimized. So basically, we have, for example, some, some cells in dataset 1 and some cells in dataset 2. So basically, we were finding the the correction vector of the total minimum length, or basically finding this so-called siblings. So our assumption that we have for each of the cells, we will have such sibling in the second dataset. So here's the simple example. Let's say we have the simplest ever scenario. We have two data sets consisting of two cells each. And so here, if we assume that each cell has a sibling, we'll have only two scenarios. So basically we'll have to bring this, blue cells by using the green shifting vectors, or we can use the second scenario as in this case. So here the ways denote the Euclidean distance between the cell expression profiles. And in the first case, we see that, you know, the total length would be basically 1 + 1, which is 2. And then in the second case, the total length would be 4. And so here, you know, like even intuitively, we see that, you know, like we would prefer using the first scenario here. Well, in this case, what we observe also is that, you know, like we're not only achieving, you know, the minimum total length, but we also don't compromise the, you know, the local structure of each of the data sets. Like in this case, you know, like we're, you know, like compromising the structure of the first batch. So now the approach, the algorithm that I'm presenting is based on the following steps. So first we, you know, like we have to identify the representative cells, since we cannot assume that, you know, like each of the data sets consists of the same number of cells. So what we do in this case, we just set the number of representative cells that we want to match. Let's say, like by default, I'm using like 100 cells. So now what we do, we PCA each data set first, right? We find principal component analysis, not in the shared space, but just in order to avoid, you know, like the curse of dimensionality, and find, you know, like a good clustering. And then we use the cluster centers as those representative cells. Now, the second step, we're building the anchor, so-called anchor graph. So that would be a bipartite graph, where obviously each partition corresponds to each of the two batches. And then this graph is a edge-weighted graph. So where, you know, like the distances, or like the weights of the edges, they represent the Euclidean distances between the cell expression profiles. Now, the next step would be computing the minimum weight matching in this graph. And so in this case, you know, like what happens, we estimate the batch effect correction vector for all these representative cells. Now, step 5 would be the extrapolation of batch effect vectors. So basically, for each non-representative cells for which we don't have the estimate of batch effect vectors, we estimate their batch effect vectors as, you know, like as a convex combination, you know, like weighted by the distance with, you know, like top k nearest representative cells in the context of the same data set. And then, you know, like estimating the batch effect vectors in each of the cells. Then we can, you know, apply the transformation by basically shifting each of the cells by the corresponding, you know, like batch effect vector. So that's the whole approach. And let me illustrate, you know, like a couple of things, how the algorithm works, and maybe contrast it with the paradigm of mutual nearest neighbors. So here I just, you know, like created some, you know, like two-dimensional, synthetic data sets, which are probably not very, you know, like not very realistic, but just to illustrate the main point here. Like here we have two batches. Let's say one is green, another one is red. And here what I'm showing, in case of Batman, these are, you know, like the representative cells that I'm using in order to integrate the two data sets. And here in MNN, so I was actually running mutual nearest MNN correct method. And here I'm displaying, you know, like I try to display at least, you know, like the mutual nearest neighbors, or like the cells that participate in that mutual nearest neighbors pairs. So here, next step would be, you know, like matching the, you know, like the representative cells. So and this is how it's done in Batman here. In mutual nearest neighbors, since there are a lot of pairs, you know, because a cell can participate in many such pairs, I just, you know, like I try to do some kind of visualization, but this is how it works here. Now, the final result, if we, you know, like use the, you know, like use these pairs to integrate the two data sets, this is how it looks with Batman, and this is how it looks with mutual nearest neighbors. So the problem here is, again, that is it undercorrects the batch effects. Now, also, if we talk about the challenging scenarios that I mentioned when I was talking about, you know, the MNN paradigm, is that, you know, like there are two challenging scenarios. One of them is when batch effects are non-orthogonal to the biological signal, and this is here. So basically, we have, you know, like these vectors here, they are not orthogonal to the biological signal. We can look at this angle here. And so in this case, what happens is that, you know, like if we have, you know, these are the weights here are Euclidean distances, right? And so these correspondence of representatives or correspondence between the cells created by Batman. And here, this would be a vector, of or the only mutual nearest neighbor if we take, top one MNN. So this would be the integration vector for, for the case of using MNN. In case we have large batch effects, like in this case, we have two batches consisting of 4 cells each, and by applying Batman, we will be able to correctly transfer these cells, and keeping their structure, translate them here so that we have no batch effects after all with mutual nearest neighbors. This is the only mutual nearest neighbor pairs, two mutual nearest neighbors pairs here. And so we see that this would be this kind of kissing effect that I was referring to in my previous slides. So this is to show that Batman is indeed able to overcome those difficult scenarios. And here a possible question that that I can get is what do we do if we have, for example, many cell types? So I was just presenting, in the case when I have 2 point clouds or 2 batches. So what happens when we have many cell types? So what do we do with this matching? So so my answer here is that I'm computing, correlations between, or let's say Pearson correlation between representative cells or between their, expression profiles. And then we filter out edges between cells with low correlations. So we are basically forbidding matching, cells between different cell types. So that's one of the things that, I'm proposing. We can, that's a parameter in the software package, but, I set it to 0.7 and it works quite well. And another problem here is that, we can have, even in the, when we match or when we want to correct batch effects between 2 data sets consisting of 1 cell type, what can happen is that our clustering may not be balanced in the sense that, for example, one cluster can be large, another one is smaller, and the structures of 2 data sets can be a bit different. So here, if we would just use, in this case, we have two batches, and if we use, the vanilla approach that I just presented, we would match this small cluster to this large one, and then this large to this small one. So that's because of, a bit uneven local structure of 2 batches. So in order to overcome these issues, I propose to, create some dummy cluster centers that would be, basically, the larger the cluster, the more fictitious cluster centers we are creating here, so that, when we find the minimum weight matching here in this bipolar graph, what we will end up with is, matching these 2 smaller clusters to this large one, and then this large one to these 2 smaller ones, so that, we don't introduce an artificial batch effect when we are solving it with, with Batman. So these are 2 things that I'm adding on the top of my method. So now quality metrics. Here, in my paper, I chose to compare Batman against other methods by using LISI. So this is local inverse Simpson index. Since such methods as K-BAT, they are not robust towards different cell type composition, and that's a known issue. So that's why I've used LISI. So LISI, it comes in 2 flavors. So basically I-LISI, which is integration LISI, and C-LISI, cell type LISI. So basically the meaning of this LISI metric is the following. Let's assume that we, we sample a cell from batch 1, and then, in the neighborhood of that particular cell, we are sampling, other cells. So LISI shows us how many cells on average we need to sample from the neighborhood of a cell in order to get another cell with the same cell, with the same batch label. So in case when we have perfect mixing and the 2 data sets approximately consist of the same number of cells, the perfect mixing would mean that we have LISI of 2. So on average, like every second cell should be of the same, like batch label, or from the same batch. But in case of perfect separation, what happens in this case is that Lisi is 1, because like we will be sampling only the cells from the same batch. So this is the metric for basically for well mixedness. And also, in order to measure the like basic the quality of our like of our local structure preservance, or how well we keep the structure of each of the two batches, I have introduced the so-called k retained nearest neighbors. So basically, for each of the data sets, we compute k nearest neighbors for each cell before integration, and then we compute the same kind of thing. Yeah. Oh, 5 minutes. Yeah, okay. And we're computing the same kind of thing, like after integration in the context of the same data set. And so we measure, we take the intersection of these two sets in order to compute how many like nearest neighbors we are preserving. So how well we keep the structure, local structure of each of the data sets. So these are the two metrics. Here I'm showing you the results, on some simulated data sets. I've used package splatter. That's basically state-of-the-art approach to simulating single cell RNA-seq data sets. And here in the original data sets, we clearly see some some separation. Here, Batman and Surratt version 3.0 were able, like at least visually, were able to get rid of batch effects. But like nevertheless, here if we look at the metrics, we see that Batman outperforms all the other methods. But visually, we see that MNN correct and Scanorama, they have some issues of like shrinking the one of the batches towards the center of the other one. That's all simulated data sets, and they might not be very representative of the real picture. So so I will talk about other real data sets. So here in the same simulated data sets, I was looking at basically like how well we like what is the Lisi score as the function of number of principal components that we're using. So visually, when we look at the pictures here, we see that like for example, Surratt was kind of able to do similar job to Batman, but that's that's the thing that it worked well only in the couple of top principal components. When we consider more principal components, we see that the quality like degrades here, and like Batman was the only method that didn't care about how many principal components we are taking in the integration. And as well, here, when I compute, like, let's say, top 10, 20, 30, 40, and 50 nearest neighbors, I see that in the case of Batman, it preserves much better the local structure as compared to other methods that completely compromise the, the two data sets. So I also use, pancreas data sets that are, that appear in almost every single cell integration paper. So here we have, I used two data sets, cell-seq2 and smart-seq2 data sets. And here we have 13 cell types. So we see that, in the real case scenario, most of the methods were kind of producing similar results. Well, in terms of, let's say, in terms of Lisi score, Batman was not the best in the top, let's say, up to 50, but then, it was kind of more or less stable. Well, what's a good thing here is that, it did not sacrifice the local structure of the two data sets. Since, if we remember the first slides when I was telling, if we don't care about local structure, we just can simply, mix the two data sets and get the best results. So finally, I also used two 10x genomics data sets. This is PBMC and PANSI cells. So
sorry to interrupt. Just to let you know that you have less than 1 minute left, so it's good.
But this is that's the last slide. Okay, no thanks. Yeah, so here we have basically PBMC cells consisting of 3 clusters and PANSI cells consisting of 1 cluster. Now, if we don't do any correction, we'll see that this cluster of pan-T cells is basically, it should be mixed with this PBMC8K0 cluster, all right? So that's basically the integration should go only here. And then finally, when we look at the results, we see that, you know, like, for example, the three methods, Batman, MNN correcting, and Scanorama, were able to correctly solve for the batch effects, with Batman outperforming other methods in terms of both metrics, while Surat in this case failed because it probably matched some cells, you know, with cells coming from two wrong clusters. So thank you very much for your attention, and I'm happy to take any questions.